Good morning, everybody. Assalamu alaikum. Today I'll be discussing my recent progress in my PhD projects. The topic of my thesis is modulating nanozyme activity for developing novel nanozyme-based point-of-care biosensors. So before digging into the details of my experiment progress, I will give a brief introduction of the different concepts that we will be discussing throughout. So biosensors, what are biosensors? Basically, these are systems or systems that can actually help detect different kinds of analytes. Analytes can be of different nature. It can be ions or biological components like proteins, antibodies, metabolites, or even pathogens and viruses. Biosensors have four major components. The first one being bioreceptor. So bioreceptors basically can be are biological in nature. It can be antibodies, enzymes, aptamers, or even synthetic receptors that we can prepare such a way that they are highly specific for the target analytes. So bioreceptors are complementary to the analyte, or they are highly specific for the analyte, so that it helps develop highly selective and sensitive biosensors. Then we have the signal amplifier component, which basically helps amplify the kind of signal that is produced upon interaction between the analyte and the bioreceptor. Signal amplifiers can be also of different nature. It can be enzyme-based, nanoparticles, or even polymers. Coming to the transducer part of the biosensors, basically the kind of signals that is produced upon interaction between the analyte and bioreceptor can be of different kinds. So if an electrochemical interaction is occurring or an electronic transfer is occurring upon the interaction between analyte and bioreceptor, an electrochemical electrochemical signal is produced. Similarly, if an optical signal is produced upon the interaction between analyte and bioreceptor, a colorimetric or fluorometric transducer is in the system. Then is the detector part of the biosensor, which actually is a surface part that helps us read out the signals that are being generated. So my work basically is focused on signal amplification part of the biosensor and how we can improve the signals that are produced. The improvement in signal or the signal amplification is particularly necessary because it helps detect target molecules in very low concentrations and allows for rapid testing, and also incorporation of signal amplification system in a biosensor helps to miniaturize a biosensor so that small biosensors can also help detect very low concentrations of the target molecules. I am particularly focusing on nanoparticles, especially those nanoparticles that have enzyme mimicking properties, or also called the nanozymes. So nanozymes are nanomaterials, as I mentioned earlier, with enzyme mimicking properties. And why are nanomaterials, such nanozymes, different from the biological nanozymes? They help us to they help overcome the limitations that are normally posed by the natural enzymes, especially concerning the stability, because enzymes get denatured easily under changing conditions, but nanozymes do not. Also, enzymes require different kinds of purification steps. Nanomaterials do not. And they do not require any stringent conditions for transportation and storage. Also, other properties, particularly surface functionalization capacity of the nanomaterial, help us tailor specific nanozymes to carry out specific reactions according to our needs. Also, they are cheaply available, they have high stability. Other properties like the optical, mechanical, thermal, electric, and magnetic properties of the nanoparticles make them particularly unique towards the development of highly specific and selective biosensors. So nanozymes can be of different types. For instance, they can be metallic if they are derived from metal counterparts, metal oxide, carbon, or even hybrid nanozymes, wherein there is a combination of metallic and non-metallic counterparts in the same nanomaterial that demonstrate enzyme mimicking properties. The kind of enzyme mimicking properties that nanozymes follow also varies. For instance, it can be peroxidase, oxidase, superoxide dismutase, catalase, or hydrolase mimicking nanozymes. So the objective of my research study basically even though there are many studies that have already been done for multiple applications of nanozymes in both therapeutic and diagnostic, I'm focusing only on diagnostic aspect, especially over the catalytic efficiency of nanomaterials for developing highly specific point of care biosensors. My study basically explores a different kind of nanomaterials, including monometallic, bimetallic, trimetallic, and hybrid nanozymes for developing highly selective and sensitive point-of-care biosensors. So each project that I will be covering throughout my PhD has 5 major components. The first one being investigating the particular nanocomposite for its enzyme mimicking properties, then optimizing the experimental parameters, assessing the performance of the proposed biosensors towards detecting target analyte, and to check whether it's selective enough in the presence of other interfering molecules, and then finally evaluating the proposed biosensors towards detecting the target analyte in a real sample. So the first project that I have covered, that I've already done, is developing hybrid nanozyme-based colorimetric immunosensor. So immunosensor or immunoassay basically depends upon the interaction between antigens and antibodies, which are highly specific interactions, biological interactions. And depending upon the kind of interaction there can be different detection signals that can be produced, colorimetric, fluorometric, or electrochemical in nature. And these signals are produced with the help of certain enzymes or fluorophores or even radioactive isotope labeled antibodies. So in my work, in this work, instead of enzymes, I have used nanozyme to amplify the signals that are produced during an immunoassay. And the target analyte model that I have used in this project is porcine gelatin protein. So the first part was preparing the nanozyme. So graphene nanoplatelets was combined with chyrosine and sonicated to produce the graphene, highly homogeneous graphene nanoplatelets and chyrosine solution, which was further sonicated with colloidal gold nanoparticles to give the nanozyme. The peroxidase-like activity could be tested in the presence of hydrogen peroxide and the colorimetric substrate, that is the TMB. In the presence of nanozyme, the hydrogen peroxide is associated to give free hydroxyl radicals, which then oxidize the TMB product to give highly blue-colored solution. The immediate color change suggested or confirmed the peroxidase mimicking activity of the nanozyme. The immunoassay was carried such that first, the nanozyme was combined with the antibody to form the antibody nanozyme conjugate. Separately, the 96-well plate was coated with a porcine gelatin protein, followed by a blocking of the non-specific sites of the well with polyethylene glycol, and then subjected to immunoassay by introducing the antibody conjugate into the solution in the presence of sodium acetate buffer and TMB and H2O2. A blue color change was observed. So if there is no protein in the well, there will be no antibody-antigen reaction occurring. Thus, there will be no nanozyme in the solution, and there will be no color change observed. That's how the system worked. The morphological characterization was done of the nanocomposite. So SEM analysis along with UV visible analysis was performed to understand the morphological characteristics. So as you can see, gold nanoparticles had highly spherical structures. Graphene nanoplatelets appeared to be thin sheet-like structure. Gold nanoparticle, the average size was 20 nanometer. And then the nanozyme had both, sheet-like structure, discrete sheet-like structure of graphene nanoplatelets, which was coated with the gold nanoparticles. Coming to the UV visible analysis, so a characteristic peak of the gold nanoparticles was observed at 520 nanometer, and characteristic peak of the graphene nanoplatelets was observed at 270 nanometer. And upon the conjugation of the nanozyme, a slight peak shift was observed, and upon further conjugation with the antibody, a peak broadening was observed, suggesting that there are certain morphological changes occurring within the developed nanocomposite. For understanding the enzyme mimicking activity, the absorbance at 652 nanometer was studied because that is where the oxidized and maximum absorbance of the oxidized TMB product occurs. So as you can see, when only nanozyme was present in the solution without conjugating to the antibody, maximum peroxidase mimicking activity was observed. And upon conjugation with the antibody, the absorbance decreased slightly, even though the color changes were significant enough to carry out further experiments. The slight decrease in the antibody suggests that the antibody covered the surface of the nanozyme, and certain enzyme active sites were covered. That's why there's a slightly reduced peroxidase mimicking activity. However, the other counterparts, that is, grapheneinoplatelets on its own or gold nanoparticles on its own, did not give significant absorbance, therefore suggesting that this developed hybrid nanozyme had the maximum enzyme mimicking properties. To study the steady state kinetics and to derive the kinetic parameters, to understand the kinetic parameters of the proposed nanozyme, Michaelis-Menten plot was plotted and Line-Weaver-Burke plot was obtained. And from the Line-Weaver-Burke plot, Km and Vmax values were calculated. So Km basically is Michaelis-Menten constant, which is an indication of the affinity that an enzyme has for its substrate. So if lower the Km value, higher is the affinity of the enzyme for the substrate. So as you can see here, it's slightly not visible, but then it's 0.048, which is about 10 times more than HRP, that is natural enzyme. So this low Km value confirms that this hybrid nanozyme has a high affinity for TMB substrate. Similarly, the Vmax value was about twice than that of HRP, confirming that it has a high velocity, the enzyme has a high velocity at a given concentration. So this experiment confirmed that the proposed hybrid nanozyme had a good enzyme peroxidase mimicking activity. Several parameters, experimental parameters, were optimized, including the nanocomposite ratio. The best activity was observed for when whole nanoparticles and graphene platelets were combined in the ratio of 1:1. So that was the optimized ratio of the nanocomposite. Concentration of TMB was optimized to 15 millimolar. Concentration of antibody conjugated to the nanozyme was optimized to 5 microgram per ml. Hydrogen peroxide concentration was optimized to 5 millimolar. The pH of the sodium acetate buffer was optimized to 4. And the time of the immunoassay was optimized to 10 minutes upon the introduction of TMB into the solution. Then after the optimization of the different parameters, the analytical performance of the proposed immunosensor was assessed, and a linear range was observed from 200 to 2000 picogram per ml of the protein. And the limit of detection was calculated from the regression plot, which was found to be 86.42 picogram per ml, which is quite low and good, and it could detect the protein with high selectivity and sensitivity when selectivity experiment was carried out. So different kind of protein were introduced into the well, and the immunoassay was carried out using the optimized parameters. So as you can see, in the presence of any other protein apart from porcine gelatin, absorbance significantly reduced. Also, interference test was carried out when only porcine gelatin was present in the well, or all proteins, including porcine gelatin, was present in the well. Good absorbance was observed. However, when porcine gelatin was excluded from the well, then the absorbance significantly reduced, confirming that the immunoassay was highly selective for only porcine gelatin protein. Then real sample analysis was carried out of different cosmetic samples and also food samples, and the real samples were diluted in PBS buffer in two different ratios, 1:10 and 1:1000, and a good recovery percentage and RST value was obtained in all the different samples. The recovery percentage ranged from 96% to 105%, and RST ranged from 1.40 to 6.71. This experiment confirmed that the proposed biosensor could be applied even in real samples and was reproducible enough in different samples of varying intensity. Upon comparison with different methods that have already been established for detection of porcine gelatin, our proposed biosensor could actually detect porcine gelatin in diverse samples with high selectivity, even though we were using only one single antibody for the experiment. And comparing with other methods, including mass spectrometry and immunochromatography and other ELISA methods, this method gave a good limit of detection and was selective and sensitive enough. Though we believe further work can still be done to improve the selectivity and sensitivity by incorporating two or more antibodies, like in a sandwich ELISA. And this study is promising enough to identify trace amounts of food allergens and adulterants in complex food samples. The paper has already been published. The study has been published in Food Chemistry Journal recently. Coming to the next project was based on fabricating trimetallic nanocomposite for detection, for developing a dual mode immunosensor. So trimetallic nanoparticles are advantageous as compared to monometallic counterparts because we have the presence of two or more metal nanoparticles within the same nanostructure. And because of the presence of two or more metal nanoparticles, there is an increased electronic transfer between the adjacent materials, which increases the enzyme mimicking properties. Studies have also shown that monometallic nanoparticles, they have a tendency of undergoing aggregation, and that results in reduced enzyme mimicking properties. Therefore, trimetallic nanoparticles are particularly promising in developing highly selective and sensitive point-of-care biosensors. So, dual mode biosensors, dual mode, as the name suggests, they have two different modes of detection within the same system, possibly. So, in this study, I proposed a dual mode biosensor, which has the properties of both optical biosensors and electrochemical biosensors, and the major advantage of dual mode biosensor is the possibility of cross confirmation of the same target analyte using the same system, and therefore it helps to confirm the accuracy of the proposed sensor. In this study, this is the basic schematic illustration. So 3 different nanoparticles, including silver nanoparticles, gold nanourchins, and platinum nanoparticles, were sonicated together in tyrosine matrix to give the trimetallic nanozyme. This like in previous study, the colorimetric detection was performed, and the same wells were washed, and the buffer was changed from sodium acetate buffer to PBS buffer, and hydrogen peroxide was introduced, and electronic charge transfer occurred that gave, and using amperometric studies, the detection was performed. So using both the methods, using the same platform, that is the same 96 well plate was used to carry out both the detection, confirming that this method was robust enough. The morphological characterization of the nanoparticles was done. TEM analysis of the individual nanoparticles was performed. So gold nanourchins, as you can see, it has irregular structure, and the diameter is approximately 50 nanometer. Average diameter of the gold nanourchin is 50 nanometer. Similarly, TEM images reveal that the spherical nanoparticles had spherical structure and average size of 30 nanometer. Platinum nanoparticles were very small and had well-rounded structures of an average diameter of 5 nanometer. And some image revealed that there is formation of the nanocomposite, wherein the 3 different nanoparticles were present in close proximity, confirming the formation of the trimetallic nanocomposite. Like in previous study, the enzyme mimicking activity was assessed by checking the absorbance at 652 nanometer, and the monometallic counterparts and the bimetallic counterparts were compared with the trimetallic, and maximum absorbance was observed only when trimetallic nanocomposite was present in the wells, confirming that the maximum peroxidase activity is only when the trimetallic nanocomposite is present in the well. Over time, also, as you can see in this graph, with increasing time, the kinetic activity of the nanoparticle increased up till 360 seconds, 6 minutes, beyond which a saturation point was reached. Also, when only either TMB is present in the well or only hydrogen peroxide is present in the well, there was no color change observed, and this is because this confirms that the material has peroxidase mimicking activity, not oxidase or any other kind of enzyme mimicking activity. The maximum absorbance was observed only when both TMB and H2O2 was present in the well. You can see the significant color changes. This is when only hydrogen peroxide is present, and this is when only TMB is present, but the maximum blue color change is observed only when both the substrates are present in the solution. Electrochemical study was also performed to check the difference between whether which trimetallic or monometallic or bimetallic counterpart gave the maximum electrochemical response. Maximum electrochemical response was observed only when the trimetallic nanoparticle was present in the wells. Steady state kinetics was also assessed, and Km and Vmax values were calculated. I'm sorry, this is supposed to be here. So as you can see, Km value was very small. The Michaelis-Menten constant was very small, almost 5 times lower than that of HRP, which confirms that this proposed trimetallic nanocomposite is highly active, and it has a good enzyme mimicking properties, and was used in further experiments. Several several parameters are optimized, including the nanocomposite ratio, antibody concentration, TMB, H2O2, and pH of sodium acetate buffer, and time. The nanocomposite ratio was optimized silver, gold, and platinum was optimized to 1:1:4, where maximum absorbance was observed. Antibody concentration was optimized to 1 microgram per ml. TMB concentration was TMB and H2O2 concentration was optimized to 5 millimolar. pH was optimized to 4, and time was optimized to 360 seconds, that is 6 minutes. Several electrochemical parameters are also optimized. The pH of the PBS buffer in which the electrochemical study was performed, that was optimized to 6. Concentration of hydrogen peroxide, that is the electrolyte used in the solution for electrochemical studies, that was optimized to 5 millimolars. And also to perform the amperometric studies, the maximum working potential of PBS was assessed using CV, and the maximum reduction occurred at when 0.7 volts of potential was applied. And so this point, minus 0.7 volts, was used to carry out amperometric studies, and analytical performance of the biosensor was assessed. So as you can see, with increasing concentration, increasing electric current response was observed, and the linear range between 0.1 femtogram per ml to 10 nanogram per ml was obtained, and limit of detection was calculated using the calibration curve, which is obtained to be 1.8 picogram per ml. Similarly, for colorimetric study, analytical performance was obtained, so that the linear range was between 1 femtogram per ml to 100 nanogram per ml, and the limit of detection was calculated to be 1.87 picogram per ml. So this, as you can see, with increasing concentration, colorimetric color changes became more prominent enough. Selectivity and interference study was also done. So different kinds of proteins were introduced into the well, coated onto the wells, and selectivity was analyzed. So as you can see, this proposed biosensor was highly selective only for osteoprotogerin, and in the presence of any other proteins, very less current and absorbance was observed. Similarly, real sample analysis, that is, serum samples were diluted in different, and different concentration was spiked. Different concentration of osteoprotogerin was spiked into the solution, and the recovery in RST value ranged for electrochemical study. RST values range from 1.5 to 2.4. And for colorimetric study, RST values range from 2.2 to 3.1, suggesting this method is highly selective and sensitive even in real serum samples. Also, the stability of the trimetallic nanocomposite was assessed. So the nanoparticle was, the trimetallic nanocomposite was stored at 4 degrees for up to 8 weeks. And as you can see, even in the 8th week, every week, the immunoassay was carried out to check the electrochemical and colorimetric response. Even in the 8th week, the nanoparticle retained up to 87.9, up to approximately 88% of its enzyme mimicking properties. And similarly, for colorimetric response, as you can see, it still retained about 95.7% of its trimetallic nanocomposite enzyme mimicking activity, suggesting that this proposed trimetallic nanocomposite is highly stable stable. Comparing to the other different kinds of trimetallic nanocomposites that have already been done, our sensor, the biggest advantage was that it allowed for dual mode immunoassay. That is, 2 methods could be employed using the same system to detect and cross-confirm the presence of the target molecule, and could perform within a very short period of time, within 6 minutes. So this is highly promising towards developing and allowing cross-conformation of the target molecules using the same system, making the process highly robust and comparatively inexpensive. I will be still covering 2 more projects during my PhD study. One is ultra-sensitive nanozyme-based CRISPR-Cas12 dual-mode biosensor for detection of pathogen, and also developing a bimetallic nanozyme for the detection of glucose using paper-based biosensor. This is my PhD thesis plan, so I've already covered these 2 projects. I still have 2 more to go. And for all the work that I have done and I'll be doing, I would like to thank the UBD Biosensor, UBD University, Brunei Darussalam, and Faculty of Science, especially UBD Biosensors, for making this work possible and giving me this opportunity. Thank you.